Okay, thank you very much, and thank you, Anna, also for the introduction. So, in the next 45 minutes, I'm going to talk about spatial domains, and as Anna already mentioned, is a concept very much related to niches, and oftentimes, even in our papers, the terms I've used interchangeably. But in the context of this workshop, we wanted to make a distinction, and Anna already mentioned it, and then I'll go also a little bit more in detail from that point of view. So, in this lecture, I'm going to first give a definition of what is a spatial domain. I'm going to show a little bit of categories of way of finding spatial domains. And I'm going to mention also our work in this context of CellCharter. I'm going to focus in particular on two interesting possible downstream analysis. One is, for example, the characterization of the shape of those domains, which sometimes have interpretable biological meaning, and also how we found the best number of domains, and also goes in the context of the question that has been asked before. And also to see the applicability, also I'm going to show a little bit of use case on how we use domains to characterize the certain diseases such as autoimmune disease and lung cancer. So luckily, we'll not have to go into the details of explaining what spatial omics is, so we can go directly to what is a spatial domain. And so in single-cell RNA sequencing, a common step that you have when you get a new data set is to cluster cells. And this allows to identify, for example, different cell types, or even looking at subtypes within the same cell type, and lets you understand what is the proportion of a certain cell type in the tissue and makes you understand what is happening. This, of course, is also done in the context of spatial transcriptomics, for example, to annotate cells. But now that we have also another information about the cells, which is the spatial locations, there is also another type of clustering that can be done, which is to cluster cells based on the cell types, or in general, the cells that you have around. And so then if you cluster in this way, you can obtain what are called spatial domains that can help characterizing the tissue architecture in the sample. And so how do you find domains? So you cluster cells based on the gene expression of the cell itself, the immediate neighbors, and this will constitute more similar to what Anna mentioned, the niche. But then you can also go further away. And so look at cells which are further away, and that's the combination of all these three components will constitute the spatial domain. So I'm going to show a few approaches, or in general categories of approaches to find spatial domains. I'll just give the intuition because there will be no time to go into the details. And the first approach is something Anna already mentioned, of looking at the proportion of cell types around the cell. And so what you will do is that for a target cell, you identify the closest neighbors of n closest neighbors, in this case, for example, 10. And then you compute and estimate the proportion of a certain cell type in the neighborhood. So in this case, out of 10 cells, 2 are malignant cells, 3 are neutrophils, 5 are lymphocytes, and so on. So you can estimate the proportion. And if you do this for all the cells in the data set, then you will obtain the proportion of cell types in the neighborhood for all of these cells. You can use this information to cluster the cells. And so you will obtain domains based on this information. This has one big advantage, is that the method is very scalable. Identifying neighbors, computing the proportion, and clustering is very, very scalable. But it depends on the manual annotation. So this raises a lot of questions, like how deep you go in the annotation. What happens if you change the annotation slightly to improve it? Then you have to recompute all the domains and so on. And also in general, how to capture the variability that you can have within different cell types, also in the context of cancer, that you can have very heterogeneous tumor cells in the same patient even. So one example of this is, for example, in the context of spatial proteomics. This is a study done on colorectal cancer, on a spatial proteomics data set of many patients. So in this case, they identified what they call cellular neighborhoods, or it could be niche or spatial domains. And in this case, what they observed, for example, is that in a specific domain, so domain 9, they could distinguish the survival and discriminate the survival of the patients based on the proportion of a certain type of T cells. So PD1 positive CD4 T cells that when they were higher in proportion in that specific domain, they led to a higher survival for patients, while when they were at a lower frequency, this led to a worse outcome. So this was the first approach. The second approach that you can see often is based on the concept of hidden Markov models. So hidden Markov models are widely used to study sequences. And so in this case, for every element of the sequence, you will have a state, which is hidden. So something that you cannot see, you cannot measure, but you will have to estimate. And then every element, which is associated to a state, also emits a certain observation. And the observation depends on the state that certain element of the sequence belongs to. And this is something that you can measure, and that is basically your data. So based on the observations that you see, you estimate the underlying states. Just to make an example, if we imagine this sequence as the genomic sequence, so every element in the sequence is a region of the genome, then, for example, the state could be the chromatin status or state, which could be active or inactive, what is known as euchromatin and heterochromatin, which is something that sometimes you cannot know. But then you have, for example, measurement on the histone marks levels. And based on the levels of these histone marks, their combination and so on, you can estimate what is the status of the chromatin. And one important concept here is that underlying this model, there is the concept of the fact that neighboring observations will be more likely to have similar states. And this serves as a sort of smoothing function that even if there are fluctuations in the levels of in this case, histone marks, this will not lead to change of states every element in the sequence. Now we think, how is that related to spatialomics, to spatial domains? I understand. So this comes from the fact that sequences in general can be modeled as one-dimensional graphs. So every node in the graph is connected to the previous and to the next one. But there is a generalized version of this model, which are called hidden Markov random fields, that can operate on arbitrary graphs. So in this case, the cells will be the nodes in the graph. The states will be the spatial domains, what you want to estimate. And then the observation would be, for example, the gene expression data that you have. And these models have the advantage of not relying on annotation. So they work directly on the gene expression data, but then they suffer in terms of scalability. So I wanted to show an example. This is a figure from the BayesSpace method, which is maybe the OG spatial domain method in which they analyze a melanoma sample of spatial transcriptomics. This is the old version before it was acquired by 10x. And they identified domains, and with the same model, they also were able to increase the resolution and refine the annotation so that they were also able to capture these smaller domains of lymphoid tissues that otherwise would have been lost in the lower resolution version. Then the third method is based on graph neural networks. So also here, I'm just going to give the intuition, but the intuition behind graph neural networks is that every cell will be associated to a vector, which is called representation or embedding. And then cells with similar neighborhoods, they will tend to have similar vectors, so similar embeddings. So this is going to be a toy example. Here, every circle, every node is a cell, and the color of the cell represents the gene expression driven by different cell types, for example. Here I wanted to show also the variability that you can have sometimes between different cell types, which are represented in different shades of the color. And if your cells are represented in this toy example with vectors of length 2, then you can plot these values in a 2D space. And so if you look, for example, in this, approximate very toy example, the two cells, for example, here, the cell 4 and 3 in red, we see that they're very close to each other because they have similar gene expression, because they belong, for example, to the same cell type. But they also have similar neighborhoods. They're both connected to a single cell here in green. Well, on the other hand, for example, the blue cell is still quite close to these red cells because even though it belongs to different cell types, so the gene expression is different, it also has a similar neighborhood, so it's also connected to a single green cell. Something that doesn't happen, for example, with this blue cell, which is connected to a slightly different neighborhood. And so also these type of approaches doesn't require annotations, work directly on gene expression, and based on the implementation, they tend to have low to medium scalability. One application, some notable mentions are Stargate and GraphST that are widely used. And for example, this figure is from GraphST, in which they computed the spatial domains for the stereoseq mouse embryo data that we have seen multiple times in these days. And they observed that many of the spatial domains they obtained were actually matching with organs or particular biologically meaningful regions of the embryo. All right, so at the beginning of my PhD in 2020, I was planning to apply graph neural networks because during the master thesis, I wanted to apply graph neural networks. Then we moved to another approach. And then when I started the PhD, we were discussing the possibility of working on spatial omics. And I found spatial domains as a very good approach and use case for graph neural networks to use for spatial domains. And because what they say when all you have is a hammer, everything looks like a nail. So I wanted to apply anywhere, and so I was very determined. And also because in 2020 there were very few spatial domain methods published, and none of them was based on graph neural networks. So I was very excited. But as you usually do, we decided to first implement a simple baseline. This is very helpful usually because it lets you get accustomed to the problem without getting stuck in the complexity of implementing the method. And also once you've implemented that, then you can start to gradually increase the complexity of what you're implementing. And so we designed the baseline that you're going to see right now, which again is based on the concept of clustering cells based on the gene expression of the cell itself, but also of its neighbors. And so, yeah, it's a little bit low, so maybe we'll have some difficulty in C. But if these are the cells here, and every cell again is connected based on the different modality that we have seen before, and every cell also has some features, which could be gene expression values. In this case, it's only 2 features per cell, but they could be more, of course. What we decided to do is that for every cell, we will do the following. We will take the gene expression of the cell itself, then move to the first hop neighbors, take the gene expression, and in this case, you will end up with multiple vectors. And so since you have multiple vectors, we wanted to aggregate it in some way. 90% of the time, we use the mean. That we have also used some use cases for other types of aggregation, but usually the default one is to take the mean. And so this will lead to a second vector. Then we will move to the 2 hop neighbors, take the gene expression, aggregate it, and so on until a certain distance L, which is usually not big. We've always used between 2, 3, or 4 hops. So then we concatenate these vectors, and if we do this for every cell in the dataset, we will obtain a matrix of cells by these spatial features. Spatial because they contain information of the cell itself, but also of the other neighbors. And then we can just do classical clustering using a Gaussian mixture model, and this will actually lead to spatial domains. And so we tried it. We tested it on a spatial proteomics dataset as a pilot, and we're very surprised because we were able to see that the domains that we were obtaining were actually matching a lot with the few annotation that we had from this publicly available dataset of the anatomy of the mouse spleen. And so we were excited at this point at a decision to make. Should I continue with the preliminary results on this method or move to the more like the originally planned graph neural networks? In the end, I opted for the first one, and from my point of view, was a good call because in a few months, then we started to see a method based on graph neural networks out, then another one, then another one, another one, another one, another one, and so on. So here I'm keeping track of the methods that have been published for spatial domains, and between 2020, so beginning of my PhD, to a few months ago when I last checked, there are more than 90. And yeah, I would say more than 50% on graph neural networks, 10% are maybe hidden Markov random fields, and then there's also other categories. So in this case, we were applying these methods. So from the gene expression, we do this neighborhood aggregation, then clustering and spatial domains, and was working well for spatial proteomics. However, there is still one piece missing if we wanted to apply it for spatial transcriptomics, because right now in this situation it would not work. So for spatial proteomics, usually you have 10, 20, maximum 100 features. So concatenating then multiple layers is not a big deal. Becomes a high number, but it's still doable. If you have full transcriptome, spatial transcriptomics, you will have 20,000. So 20,000 plus 20,000 plus 20,000 plus 20,000 will become 80,000 features, and it's clearly not a good idea to cluster 80,000 features. So the solution was just to add dimensionality reduction step here before. And in this case, for example, for spatial transcriptomics, we decided to use a CVI that scales well and gives very good results. And in theory, can also be used for removing batch effects. So it's a double solution for two problems. And so in the end, these methods, we call it CellCharter, and it's particularly for us useful because it's very scalable. So we analyze the Murphy's dataset on more than 8 million cells in less than 15 minutes in a GPU. And also this method doesn't require annotations, but works directly on gene expressions. Then, of course, doing annotations helps as a way to interpret your niches or domains. But in general, it doesn't influence the results of your domain method. And this, for example, is a figure from, not our paper, from Niche Compass, which is a more recent method to find spatial domains. And we can clearly see that CellCharter scales very well with the dataset size. This is more than 200,000 cells, and with running in few minutes compared to other methods that either fail or take longer. But we didn't want to limit ourselves to identifying domains, but also we developed a sort of framework to characterize and compare those domains. So one could, for example, look at simply the proportion of the domains in the sample and compare them between different conditions or associate them with patient survival. One could characterize those cells by looking at which cell types they are composed of and which cell types are enriched compared to random chance. Looking at the relative arrangement of those domains, if domains that are always found to be close to each other in one condition, then they're found to be far apart in another. And something we're very excited about, which is the fact of characterizing shapes of domains, which, as I said, sometimes have biological meaning. So we'll go a little bit in details on the shape analysis. So for the shape analysis, we developed 4 metrics to characterize shape. One is curl, which measures how twisted or curl a certain domain is. Elongation or aspect ratio, which is the ratio between the major axis and the minor axis of the domain. The linearity, which measures how much a certain domain can be approximated as a linear path, and regardless of whether it's a straight line or a curved line. And then all the domains, to identify the shape of these domains, we always draw automatically a boundary around the domain. But still, there could be cells of other domains within the boundaries, and purity measures exactly that. And we show that by combining these different metrics, we were able to distinguish between different shapes of the domain, like linear, round, circular, or more irregular shapes. This is to go more in details. So what we do initially is that for every domain, we split it into different components. The reason why we call them components is because we find simply the connected components of that specific domain in the graph. And so after that, we automatically draw the boundaries of these components using a concept called alpha shapes. And then we compute the shape matrix for every component so that we can then compare. And we applied it to the spatial proteomics dataset that I've shown before. This is a publicly available dataset of 9 samples of mouse spleen, 3 of which are healthy mice, and 6 are mice with the lupus autoimmune disease. And what we saw at the beginning is that actually with the progression and the emergence and the progression of the disease, there is no new spatial domain, meaning that there is no combination of cell types that is unique and appears. But these domains, certain domains tend to expand and other ones tend to reduce. I will go more later on the details on the interpretation of those domains. But what you also observed visually is that this purple domain here started to infiltrate inside the orange domain. And this made also the orange domain more irregular. Oops, sorry. And then we also looked at the domains that represent boundaries, like this light blue one and this purple one, that through the emergence and progression of the disease, they started to lose their boundary shape, and so linear or circular. We were able to quantify that. And so, for example, this boundary, this purple boundary that I just mentioned, increased in curl, meaning that it became more irregular, and also lost linearity. We can, for example, see it here between healthy spleen and lupus. This orange one also became more irregular, and so this is reflected in a higher curl. And this marginal zone here, which is still a kind of a boundary, it loses linearity and gets kind of disrupted. And so when we interpreted also what this domain represented, so this orange domain are germinal centers that in the mouse spleen are areas full of B cells, follicular dendritic cells, and in general, its role is to make B cells mature and get selected. And then this purple one is a boundary between the germinal centers and the PALs, PLAS, which are enriched in CD4 and CD8 T cells. And so this makes observe that, as expected, what happens in the autoimmune disease actually resembles what happens in a real infection. Because the T cells, the one at the boundary between the germinal center and the pulse, starts to infiltrate inside the germinal centers to activate the B cells and activate the immune system in general. So it was very nice to see. Okay, then before we move to the last biological use case, wanted to go a little bit on a digression on how we found the best number of clusters, because this is also something that was mentioned in a question before. Because it's also something that is sometimes hidden or overlooked in many studies. So in our case, our approach was based on the concept that there may not be an equivocally best number of clusters. So as we mentioned before, tissues are often organized in a hierarchical fashion. So in the case, for example, of cancer, you can have cells which are categorized as tumor cells. But then, as we well know, tumor cells of different patients are actually very different and very heterogeneous. So one could characterize them at the level patients, and they will be different. But one could also characterize them at different subpopulations within the patients, because there is often a strong heterogeneity even within the tumors, in the tumor cells within the same patient. And there is no true or more correct level of representation. All three are equally valid, and all three must be explored, but it depends on the purpose of your study. And so this was the principle behind our approach to find the number of clusters. And so was to find one or more good candidates within a range that is defined by us between K-min and K-max. And so this approach was based on what we call stability, which means how similar are two clustering results when you change slightly the number of clusters K. So in this case, you have the results between using 5 clusters or 6 clusters are fairly similar. So we assume that these results are fairly stable because maybe it's less subject to noise, or in general fits well the data. And so the way we do it is that across all the possible options in your range, you will run clustering for each one of these options, and then you will measure the similarity between the results at k and k minus 1 and at k and k plus 1. If you do this for all the values in the range, you will obtain a curve, so one value for each of these, and then you can compare them. But also these spatial domains method, they tend to have quite variability and stochasticity. So even with the same parameters and the same data, you can have slight variation of the results. So in this case, to overcome this problem, we decided to run clustering multiple times for each of these three different levels of granularity, let's say. And then what you will do is that you measure the average similarity between all the combination at k and k minus 1 and k plus 1. And this leads you to a curve that we call stability curve with peaks, and every peak probably is results of a very stable solution, let's say. Of course, this method is not for all clustering methods. So we designed it because CellCharter is particularly scalable. 90% of the time that is spent in CellCharter is dimensionality reduction. So clustering takes few seconds or few minutes. So repeating that multiple times is not a huge problem. But of course, with more expensive clustering algorithms, this clearly is not the best approach. So we applied this on a data set of lung cancer, which is a recurring topic in our lab. And so this is a spatial transcriptomics dataset from the CosMx technology. These are 8 slides from 5 different patients for a total of 750,000 cells. And this technology measures a panel of 960 genes. When we compute the cluster stability, we had peaks at 3, 8, and 20 clusters. And so our next question was, let's look at what those domains and what they represent at these 3 levels of granularity. So when we looked at 3 clusters, we saw one domain here in blue that spanned all the tumor areas from all the patients. Now what I meant here, these are domain. So this domain contained all the tumor cells, but also other cell types that co-localize with the tumor cells. And when we moved to a more fine-grained view at 8 clusters, every patient's tumor was split into a separate domain. And at 20 clusters, we detected subdomains that were composed either of different tumor subpopulations or different combination of tumor cells and other cell types. And after that, we focused on a single patient, which is patient number 9, because it gave very interesting results. So when we looked at this patient, we identified 2 domains composed almost exclusively of tumor cells, which are the pink and the brown one. This, for example, shows that using directly gene expression leads to more easily solutions like this, because using the cell types, maybe we would have not noticed that there were 2 tumor subpopulations in this patient. We may have annotated all these tumor cells as simply tumor cells, and then we will not have gotten 2 domains. And this is because these 2 subpopulations of tumor cells are actually very different transcriptionally. One population, the pink one, is undergoing response to hypoxia, which means the lack of oxygen. And the other subpopulation is more proliferative. So this shows a bit the advantage of using directly gene expression in this case. But what was also interesting is that those 2 subpopulations were surrounded by different other domains. In particular, whenever you saw proliferative tumor cells in brown, you will see a domain around composed of mainly tumor cells and CD4 memory T cells. And whenever you saw pink hypoxic tumor cells, you would see nearby a domain composed mainly of neutrophils and natural killer cells. Although other cell types, but these were the main 2. And this last association, the one between hypoxic tumor cells and the neutrophil-enriched domain, was the most striking because we were able to observe it also in our 2 large-scale spatial transcriptomics datasets and spatial proteomics datasets of lung cancer, meaning that this was not something that was simply specific for that patient. And also we started to see some hints from many bulk RNA sequencing datasets of lung cancer. And so I wanted to show this analysis because it shows that through the use of spatial domains, we were able to characterize two types of heterogeneity within the same patient. Heterogeneity of the tumor cells, because we saw two different subpopulations characterized by different transcriptional states, but also surrounded by different domains, and so surrounded by different microenvironments. Of course, we still don't know which causes which. If the changes in the transcription of the tumor cells induces changes in the microenvironment, or vice versa. And so to conclude, today I wanted to give a brief overview of what spatial domains are, the different approaches that one could use to identify domains, and then in particular on CellCharter, a way to find the approach that we used to find the best number of clusters and the analysis on the shape of those domains. But in general, I wanted to show how domains can be used to characterize the tissue architecture of samples, and also to understand how this tissue architecture gets remodeled with the emergence and the evolution of diseases. And that's it. I'll be happy to have questions.